Hello students, welcome to the biology class. Today we are continuing with our chapter Biomolecules and our today's topic is enzymes. This is another important part of your biomolecules. What are enzymes? Enzymes are proteins that function as biological catalyst. What do you mean by catalyst? Catalyst is a substance that speeds up a chemical reaction but isn't changed by the reaction. Catalysts only participate in the chemical reaction. They increase or decrease the speed of the chemical reaction, but itself there is no change in the catalyst. And enzymes catalyze all aspects of cell metabolisms which are taking place in the living organism. They are participating in carrying out the various metabolic reaction of the living organism, hence known as biological catalyst. Characteristics of enzymes. Enzymes are proteins that catalyze the biochemical reactions in the living cells and hence called as biocatalyst because they are participating in the biochemical reaction of the living organism. They are specific for their substrates, that is, each enzyme catalyzes the reaction of only one substrate. And this is due to the configuration of active site. Configuration means the shape of an active site of the protein. Each enzymes require specific optimum means variable pH and temperature. So some enzymes they act as the acidic pH, some they are variable at the alkaline pH. Each enzyme accelerates the reaction by reducing the activation energy. Catalyst and activation energy. For any reaction to take place, the reacting molecules must achieve a higher than the normal energy. And this higher energy is known as activation energy. Until the reacting molecules attain the level of activation energy, they do not interact. Hence, the chemical reaction does not proceed. If they are not getting the high amount of the activation energy, no reaction can take place. But the activation energy of chemical reaction means the requirement of activation energy of chemical reaction can be lowered by certain substances called as enzyme, that is they act as catalyst, which are required in small amount. This means enzymes lower the energy barrier for chemical reaction and permit the reaction to occur at normal body temperature. After complication of the reaction, the enzyme releases the products and free energy difference between the reactants and the products remains the same as of a activation energy of enzymes. If enzymes do not play this role, then the energy of activation will increase and the reactants means the reactants of the chemical reaction, they cannot cope up with such increased amount of activation energy and then they fail to react in the living system because they possesses insufficient kinetic energy to overcome the energy barrier for the reaction. So in this graph, you can see the reactants, they needs a very high energy, that is the activation energy, and they are unable to cope up with this large amount of the increased activation energy. So they cannot interact, they cannot react, and they cannot give the product if they requires the large amount of the energy. But with the participation of enzyme, the requirement of the activation energy is decreased or lowered and the reaction can proceed and the products are formed under the favorable conditions. Chemical natures of enzymes. Structurally based enzymes are of two types. One is simple enzyme. Simple enzyme means it is formed of only proteins and no other group is associated with it. Every enzyme is protein, but whereas every protein is not enzyme, because the proteins, they are having many other functions like transportation, like hormone formation, like formation of antibodies, but all the enzymes, they are protein in nature and they are very specific in their actions. For example, pepsin and trypsin, which helps in the digestion of the food in the living cell. Conjugate proteins or the conjugate enzymes are there. It is a form of protein part as well as the non-protein part. Means the conjugated enzymes, they are formed of protein part as well as the non-protein parts. So that's why they are known as the conjugated enzymes. The protein part of the conjugated enzyme is known as apoenzyme and the non-protein part of the conjugated enzyme is known as cofactor and they form a bond with each other. So this space, this is known as an active site which are very specific with the substrate. They form the bond and then the structure is known as the holoenzymes. So conjugate enzymes consist of two parts. Apoenzyme, it is a proteinaceous part of an enzyme which is loosely or tightly bound with the non-protein part depending upon the non-protein part. Cofactor, it is the non-protein part of a conjugate enzyme. The cofactor makes a protein catalytically active. Apoenzymes, that is a protein part, plus cofactor, that is your non-protein part, they link to each other to form a functional enzyme that is known as holoenzymes. Cofactors. There are three types of cofactors and they are prosthetic group, coenzymes, metallines. What are that is cofactors, which is the non-protein part of a conjugate enzyme. They are having essential ions as the metallines which are tightly bound with the apoenzyme and the coenzymes, that is co-substrate or the prosthetic group, they are also tightly bound with the protein part of the enzyme and they are known as the non-protein part of the enzyme. Prosthetic groups. These are the organic compounds that are tightly bound to the apoenzyme. The example is heme is a prosthetic group of enzymes peroxidase and catalases and form the part of the active enzyme. Coenzymes, these are the organic compounds that are bound to the apoenzymes only during the course of catalysis when they are participating in the reactions. Their association with the apoenzyme is only transient, means changeable. Example is nicotinamide adenine dinucleotide, that is NAD, nicotinamide adenine dinucleotide phosphate, which act as the energy currencies or the energy cells of our bodies. Metallines. Metallines forms a coordinate bonds with the side chain at the active site of the enzyme and also with the substrate. The example is zinc is a cofactor for carboxypeptidase and the carbonic anhydrase. So these are the non-protein parts which form with the protein part, that is your apoenzyme. The combination of apoenzymes and the non-protein part is known as the holoenzyme, and enzymes, they are protein in nature as well as sometimes they are having a non-protein parts to be more effective catalyst because they helps in speeding up the chemical reaction, that is a biochemical reaction of the living cell, and hence they are known as your biocatalyst. So next lesson on the enzyme classification. Till then, stay happy, stay home. Thank you and have a nice day.